I think the TATTON results are very exciting, and I'll tell you why. We've been using EGFR inhibitors in lung cancer for 20 years, and targeted therapy has truly transformed the way we treat this disease. However, personally, and I think the community would tell you this too, no one's ever cured because the tumors continue to become resistant to targeted agents. And even though we're in our third generation of drugs right now, osimertinib, which is the most specific and targets T790M, which is one of the most common forms of resistance, now we're seeing other forms of resistance develop. So we need to stay ahead of the game and find ways to treat these patients better. And the TATTON trial, and it's only a partial report, is showing two of the arms of the TATTON trial, those patients who have c-MET upregulated on resistance and those patients who don't. For those patients who have c-MET upregulated, they get a MET inhibitor, savolitinib, and for those who don't have any MET, they get a MEK inhibitor, selumetinib. The results, I think, are quite compelling for the MET inhibitor, and you would expect that to be better because those patients are selected for MET by a number of different methods, the primary one being FISH, fluorescence in situ hybridization, showing multiple copies of the MET gene, so you have more MET protein. And it looks like that in patients either who have had first or second generation EGFR inhibitors who are T790M negative, or those who have had third generation drugs, meaning osimertinib or some others, the response rates are quite high. It's still very early, but the response rate and duration of response look good enough that I think it needs to go immediately to a phase 3 trial, which are already ongoing or planned. So I think this offers great hope for patients with c-MET, which is upregulated. And one thing that we're finding is this bypass pathway, and now that we're using agents that target T790M, seems to be upregulated at rates above 15%. Some even think it's close to 30%. That's something we'll learn more of as we learn how to better detect it. So that was very exciting. As for the MEK inhibitor, it's hard to say. That's an unselected group of patients. The results are much earlier. My sense is that since you're giving osimertinib plus the selumetinib, many of the patients who are responding would have responded to osimertinib alone. But there might be something there, and I think it's worthy of future study. But a very good effort, to really find the doses for both drugs and combinations with osimertinib to show that this can be used in patients with refractory disease, and to really keep pushing and pushing to try to find ways to extend the life of patients with EGFR mutated disease as long as possible.